All right, thank you so much. All right, so as she said, I'm John Arigo. I'm talking today about improving the sterility assurance application to the FDA. So the way I have this set up, let's give you a little background. I've asked managers, reviewers, a bunch of people in microbiology for the last 10 plus years what they see as the most common problems in applications. Then we all discussed and we thought, okay, these are the big things, I'm going to talk about them. We're going to keep trying to hammer this home so we can improve the submissions. So the first thing, it might seem obvious, but I think this is worth it. Make it easy to read. So, quick story, I like to ramble. When I left grad school, I ended up in a patent law firm. And one of the senior attorneys said to me, when you're trying to make a point, say what you want to say in the beginning, say it again in the middle, and then say it again at the end. Now that doesn't exactly apply here, but it's something I've never forgotten. And if you use a little bit of that tip, you're going to get your point across. Because remember, a reviewer is sitting there and they're overwhelmed with all of this information. So make it very clear. So let me talk about this like an autoclave qualification, for example. Look at the sentence I have written here. Autoclave X was qualified by performing 3 empty chamber heat distribution runs, 3 worst case heat penetration biological indicator runs in 2016. The worst case load covers all loads proposed for production. Results are provided on page 18. Sometimes we just see results, and there might be hundreds of pages of results. But look at what that sentence does. It tells you what autoclave you're talking about, what types of runs were performed. It tells the date. Now this slide's a little old, so 2016's not the greatest, but you get the point. It talks about what types of loads, worst case, what it covers, and it tells you where to look for the load, the actual results. You can also provide summaries of the results. Tell us what the minimum and maximum temperatures are, the minimum maximum F0 and the BI results. You can say that the summary is on page 15, the actual results are on page 18. That really can move the reviewer along. And give the simple things. Which machine are you talking about? What autoclave? What is the name? What is the filling line name? Are there multiple names that you use for this line in your facility? What room numbers when you're talking about it? For example, don't assume that we know there's only one filling line in a building. That might be common sense to you because you know, well, this is the building we work in, there's only one filling line here. But we might not see that right away. So by calling it, oh, the filling line has been qualified, we might think which one, because we're used to seeing facilities that have multiple filling lines. Certainly English translations are critical, and all of this is to move the reviewer along for a faster and more efficient review. So as I said, I'm going to jump topics quite a bit here. So now sterilizing filtration. We see quite an increase in the use of pre-sterilized, commercially available filling and filtration trains. That includes pre-packaged, pre-sterilized filters, tubings, tubing, large flexible bags, et cetera. So this is all great, but just clearly mention the use of this in an application, right? Where is it being sterilized? Are you purchasing it sterilized? If you're purchasing it that way, are you relying on a DMF to provide the sterilizing data? If you're sterilizing it yourself, tell us that. And I'll talk a little bit about DMFs later. But the idea here is it just doesn't hurt to tell us too much information about what you're expecting us to quickly understand. We might not pick this up. This is trying to avoid deficiencies, right? So if you're buying the filter sterilized, you might know that it's in an autoclave load, but we didn't know it's in the autoclave load, so we're trying to find a DMF because you've sterilized it as well and you've put it in the autoclave load. If you put it in there and we've already looked at the autoclave load, then the problem's solved. We might not know if your worst case load covers everything, so tell us exactly what you're trying to do and what you're trying to accomplish. We also see reuse of sterilizing grade filters during production campaigns. We recommend you avoid doing this, but we understand that you can. So when it's possible, when you're trying to do this, just tell us the maximum number of times you're trying to reuse it or re-sterilize it, because that way we're going to need to see the data to support that. We don't want to review some data and then find out down the road that, oh wait, they're planning on reusing this 5 times? That means that we have to go take a look at this data again, and like for the bacterial retention studies, did it cover 5 uses? That's the kind of questions we're going to be asking. So we want to see that the data utilizes a worst case scenario for the amount of times you're reusing it. All right, so drug master files. I think we all probably know what this is, but they allow confidential information to be referenced from another party. However, when it's not appropriately referenced, DMFs tend to cause a lot of confusion, and they can decrease review and application approval efficiency. So I've used this picture down here for quite a while, but this was back in the day when there were large volumes that the reviewers had to literally walk across the street to a document room, check out 5 or 6 or 10 volumes, put it in a cart, and push it through a parking lot back to your office. Now everything's electronic, but I'm still scarred from those days. So that's why I want this thing here, so we never forget that. But the recommendation here is clearly tell us where the information is. These DMFs can be enormous. These companies are huge, and they have an enormous amount of data in them covering many types of products. If you're using it for a rubber stopper, they might have tons of rubber stoppers, different sizes, different chemistry of the stopper. And just saying, hey, go look at DMF 1, 2, 3, 4 can be an hour or more to try to find what we're talking about, or what you want us to go see. So if you're relying on a DMF, please ask the company where the exact data is for what you have purchased and what you want us to review. Because we have to issue deficiencies if we don't know where it is. We can't just dig through a DMF forever, because it just takes forever. And again, that's the theme here. We're trying to increase the reviewer's efficiency, which essentially speeds the approval process. All right, so most of this I'm going to talk about sterile situations, but in this scenario, we've come up with this situation where the type of product is a non-sterile powder, and the non-sterile powder has package insert instructions to reconstitute in an aqueous solution, and then there's a hold time associated with it. So the two situations I'm going to talk about is a multi-dose scenario and a single-dose scenario. And the single-dose scenario has an excessive hold time. So again, we have a powder as the product, then the instructions say mix with an aqueous solution, and then you can hold it 30 days, 7 days, whatever that may be. And our recommendation is this. For a multi-dose scenario, we want USP 51, the antimicrobial effectiveness test. We want USP 60, Specification testing at release, or a risk assessment, right? So I think that's key there, or a risk assessment. If you don't want to do USP 60 at release, that's okay. But then show us how you've controlled your water systems, how you've controlled your manufacturing environment for Specification, that you're looking for it. Prove to us, explain to us that you've done an assessment of this, that your facility is free of this organism, and then you won't have to show it at release for every batch. We also want an assessment of preservative content at the end of the storage time, because some of these products can be held for 45 days. So show us that the preservative is still there at 45 days. So now the single dose scenario. This is when the package insert has excessive times for a single dose product, and it usually does not have a preserving agent in it. We're looking for a microbiological challenge study over the proposed storage time. So this is when you can put your microbiology hat on and design a study. What organisms do you want to use? You're spiking it with an appropriate amount, and you're carrying it over the storage time, and you're going to show us that no excessive growth has occurred. In addition to that, we also want USP 60 for the Specification at release, or you can do a risk assessment. That's the key here, because these are aqueous solutions, or they're being resuspended in aqueous solutions. We want to make sure that your manufacturing facility has controlled for Specification. And we think this is providing an improved risk assessment for these types of products. All right, bulk bioburden sampling. So this is when you're sampling the bulk solution to see how clean your scenario is, right? So sometimes we see situations where there's a 0.45 micron pre-filter, there might even be a 0.2 micron pre-filter, and then there's a sterilizing filter. Sometimes we see the sampling of the bulk solution taking place in between these filter setups. And that's really not acceptable, because these filters they work well. So if you sample in between a 0.45 or a 0.2 micron filter, it's going to show no organisms. What we want is for you to understand that and do the testing prior to any filtration. So sampling after a filtration doesn't tell you how well controlled your solution is. It can give you a false impression. And certainly microbial metabolites can pass through the filters, right? Endotoxins can come right through it. And as it says, it's going to give you a false impression. You're going to think that you're controlling the solution, but it's just because you've sampled after a filter. So we'll issue deficiencies sort of asking, where are you sampling this? And if you're sampling it in between a filter, to change the sampling to prior to filtration. All right, biological indicators. So these are going to be used for autoclave qualifications, right? These are the spore-forming organisms that you're trying to see. Does your high heat really kill these? And what this is focused on is the incubation time. And the idea is 7 days or an incorrect 24-hour incubation. So sometimes we see 24 hours, 36 hours, but what we're looking for is 7 days. This comes from USP 55, which suggests 7 days after inoculation. The ISO document, commonly recognized to be 7 days. So I'm providing this here just to show that 7 days isn't something that we're just making up. This is the established 7 days. But what happened here, and no one's at fault. CDRH is not at fault for doing this. This is just, this guidance has come about and people have misinterpreted it. So CDRH issued a guidance for healthcare facilities, right? So it's for sterilization systems in healthcare facilities, hospitals, dentist office, things like that. And you can go read this at your leisure, but I'm trying to just point you to the yellow here. They're saying the incubation period for BIs can be reduced from the standard 7 or more days, provided you have validation studies, right? Now, the next bullet down there says if you have 97% of your BI positive control grow out. So they're getting this. If you can recover 97% of your positive control at 3 days, then they're saying that's okay. So for the drug world, for the manufacturing facilities, we say that's not okay. And the reason is because of what I said. It's not a complete grow out, right? Is 97% of a positive control, is that acceptable for your facility, for your manufacturing facility? No. So please do 7 days. All right, so Erica's going to talk about endotoxins next, but one thing that I have in my slide here that we commonly see is just the idea of pooling of samples, right? So USP 85 in the question and answer section of that, the addition to that document, says that you can pool 3 units. So rather than do 1 test, you can mix 3 units and then, rather than do 3 tests, you can mix 3 units and test once. But what happens here, if you think about what you've just done, you've made a 3-fold dilution. So while you're doing your endotoxins math and you're calculating the maximum valid dilution, you need to adjust that. So you take your maximum valid dilution and divide it by 3 if you're pooling by 3. Right? You don't have to pool, but what we notice is in the instructions on how to perform the test, when we're seeing how that's written, it might say, oh, pool 3 vials. But then the MVD has no divided by 3. And the reviewer's thinking, well, wait, did they account for this? Are they aware that basically they're diluting an extra 3-fold? And this can directly interfere with the ability of the test with inhibition and enhancement and things like that. So please consider that pooling concept. And if you are doing it, please tell us and then adjust for it. I threw a bonus down here. So while you're looking at the package insert, think about the pediatric dose. A lot of times we see the pediatric dose being missed, right? It's not all about the adults. There might be infants, there might be children that are being prescribed this medicine, and you need to adjust for that. There's usually different doses and things like that. So keep your eye out for that. All right, media fills. We want you to clearly indicate the maximum proposed time for production, the filling time for production, right? So we'll see these beautiful media fill studies, and they might be for 18 hours, 20 hours, but there's no mention at all of how long you're proposing to fill for production. Well, if you're proposing, I don't know, 36 hours for production, well then, are your media fills really worth it, anything? Did you prove to us that you can fill for that much time? So clearly indicate what you're proposing for production, that way we can compare it. It seems quite straightforward, but I think there's can be a mix-up into what you think you need to tell us. And if lyophilization is utilized, let us know exactly what lyophilizer was used in the media fill. Right? This really goes for any piece of equipment that you're using. The media fill needs to include the exact equipment that you're proposing for commercial production. And this gets back to that early slide I had where I'm asking, just tell us over and over again what you're using. If you have 4 lyophilizers that you're proposing to use, then we want to see those lyophilizers to be incorporated in the media fill. Tell us the names of them. Tell us which filling line. Tell us again what filling line it is, just to make sure we know exactly which one you're trying to qualify. All right, so now I'm moving into supplement filing tips, and it's the same theme, right? Tell the FDA, tell us. If you're proposing a bunch of changes, just make that cover letter so crystal clear in what you're trying to do, right? Even if you have to bullet, change number 1, we have a new rubber stopper. Change number 2, there's an increased filling duration. Change number 3, there's a new autoclave load, right? Just make it so clear on the front. Don't assume that we have all the prior reviews and submissions easily accessible. Right? As wonderful as technology is, we all know what Adobe can do when you're trying to open a PDF that might be 600 pages, right? It can be very painful. So if you just have this very clear in the front, You can tell us what we're looking for, and we don't have to go into the historical records of these reviews, which might be over 10 years old. We might not even be able to find them because they're in a different repository, something like that. So try to make it clear, right? We have a new rubber stopper, we're changing from this size stopper to this size stopper, or it might be something simple. We're simply changing the composition of the stopper. There's no change to the size. Just things like that. Tell us there's no change for things, really helps. If you're doing like a prior approval supplement, trying to get to CBE 30 or CBE 0, I think the best thing to do is just simply tell us, what are you basing it on, right? Tell us when and what was approved in the past. If it's not a confidential issue, and you can tell us, hey, ANDA number X123 in January 2023 was just approved with this same filling line. I mean, that is a huge time save to the reviewer. And then make it logical, right? We propose CBE 30 because this filling line was previously approved on January 1st and nothing else is changing, right? I added the italics there because I think the reviewer would just love to see that phrase, nothing else is changing. Because it sounds crazy, but when you're sitting there reviewing it, it's so easy to think, are they changing this? Why did they give me this autoclave data here? It's supposed to be something different, but there's all this data. Should I review this? Why do I have to review it? Maybe they're just giving me too much information. But if nothing else is changing, maybe you just gave that and we don't really need to see it. So therefore the reviewer doesn't have to waste 3 hours. So think of it in terms of that. All right, time for the quiz. This is intense. So if your manufacturing plan includes purchasing a pre-sterilized filter, but you also put that filter into the autoclave load, what do? Do you tell FDA you're also sterilizing it yourself and include the data? Do you tell FDA you're sterilizing it yourself and do not include the data? Or you don't even tell FDA anything about this? Any thoughts here? I know it's the afternoon. All right, hey, I'll give it to you. Tell us you bought it sterile, include the data, give us a summary, do everything you can to make this easy for us to review. If we don't have to dig into doing a DMF, we don't want to. We've already reviewed the autoclave, right? Remember that, we've already reviewed the autoclave data. If you've got the filter in a worst case load, we're done. If you don't tell us that, now we dig into a DMF and you're picturing me 10 years ago pushing the volumes across the parking lot. All right, question 2. You're sampling the bioburden of the bulk solution and you're sampling it in between 2 filters. Is this okay? No, there we go. Yeah, she's good over there. No. All right, so the next couple slides, I have just a bunch of references. You're welcome to look through those. There's my contact information, and up next is Erica, who's going to dive into endotoxins a little deeper.
Oh, thanks, John. You set me up nicely for some stuff. All right. Good afternoon, everybody. My name's Erica Filer. I'm here on behalf of the Office of Pharmaceutical Manufacturing Assessment, and this afternoon I'm going to talk to you about the bacterial endotoxin specification. All right, couple of learning objectives for us today. So, what are endotoxins and why are they a patient risk? How do you control endotoxins in injectable drug products? And how do you go about setting the acceptance criteria for the endotoxin specification? Let's jump in. All right, so why am I here? And don't worry, this isn't a meaning of life question, but rather why has SBIA dedicated a session at this conference for me to talk to you about endotoxins? Well, first of all, the people in my group are the people who are scrutinizing the endotoxin specification found in your ANDAs. It's something that we scrutinize very closely because we know that too high a level of endotoxins in the drug product presents patient risk. And finally, we get a lot of ANDAs with the endotoxin specification set too high. And so this necessitates us to send information requests. We may have to ask an applicant to change a specification. They may need to do new methods suitability studies, stuff that we want to try to avoid during the assessment cycle. We also get a lot of questions about the endotoxin specification through our control correspondence program. So, taken all together, this is an aspect for which there is patient risk, and there seems to be a lot of confusion on the topic. So I'm here today to tell you how we approach looking at that endotoxins acceptance criteria to try and help you get that specification set right the first time when you submit your ANDA. Okay, so what are endotoxins? Now, endotoxins, they're just a normal component of the cell wall of gram-negative microorganisms. Now you see I've got a little cartoon here on the slide, and the cartoon is a cross-section of a gram-negative microorganism, and you see those little hairs there on the top of it, those little red molecules labeled lipopolysaccharide. It's just another name for endotoxin. Now these molecules can actually have a very dramatic reaction with the human immune system. So the reaction they can have can lead to outcomes such as fever, shock, and if you're exposed to too many, even death. And so, this is something that we want to scrutinize very closely. Now, this is not a theoretical concern. There have been noted instances of endotoxin reactions to injectable products, including one notable occurrence involving gentamicin in the late 1990s. So, this is something that we at FDA and you in the parenteral drug industry are keeping a very close eye on because we want to prevent that type of adverse patient outcomes from our drugs. Okay, so microorganisms are ubiquitous in the environment. That's a fact. So how do we go about controlling the presence of endotoxins in our drug products? Now, I bet some of you are thinking, well, Erica, come on now, the drugs are sterilized, right? And that's going to get rid of the microorganisms. That's correct. Sterilization gets rid of live microorganisms, but endotoxins, they're kind of the remains. They're kind of what's left over also from killed microorganisms. So all the microorganisms that the product is exposed to along the manufacturing path, they can have an additive effect on the endotoxin burden. And as it turns out, most common sterilization methods do nothing to inactivate the endotoxin molecule. So sterilization isn't going to help us here. So what can we do to reduce the risk of endotoxins in the drug products? Well, this is an aspect of manufacturing for which we have to take a quality by design approach, right? So for the typical quality by design approach for small molecules to keep endotoxins at safe levels, it's going to involve activities like choosing the correct grade of API and excipients for the drug product. Going to want to keep good bioburden control, particularly at the formulation stage. And I know John talked about that just a little bit in his presentation. We're going to want to be sure to depyrogenate our container closure system, get rid of the endotoxins where we can. And finally, the cornerstone of any good mitigation strategy is going to be some in-product testing. Now, I've highlighted release and stability testing here on this slide. Now, why did I highlight that? Because that's where we see most of our issues in our applications. So let's take a closer look at that specification for endotoxins for our drug products. So we know that a specification consists of a test, a method, and an acceptance criteria. Let's break it down as it relates to endotoxins. So obviously our test is going to be for endotoxins. The method that we most commonly see for ANDAs, for endotoxins testing, is found in the United States Pharmacopoeia's Chapter 85. Now, this is also a harmonized chapter, so you're going to see this in the European and the Japanese Pharmacopoeia as well. And within the chapter, there are a number of different methods that you can choose from to perform your endotoxins testing. So we got our test, we got our method, our acceptance criteria. Now, the acceptance criteria for endotoxins, that can also be found in USP 85 as well. Let's take a closer look at that. Okay, that's it. That is the acceptance criteria for endotoxins in sterile injectable drug products. It makes you do a little work, doesn't it? It makes you do a little bit of math. But it's pretty simple, it's pretty straightforward. So to get the endotoxins acceptance criteria for your drug product, you have to divide the variable K, which represents the threshold pyrogenic dose of endotoxin per kilogram body weight, and you have to divide that by the variable M, which is the maximum recommended bolus dose of product per kilogram body weight. Okay, I mean, it asks you to do a little math, but it's not that much, and it only asks you for two variables. So I guess the only question remaining for this is going to be, what are K and M? Okay, so here at this conference, I am here today to tell you the secret to finding K. It's really hard to find, actually. So on the screen, I've got a snapshot of the last page of USP Chapter 85. It's small, but I'm just trying to show you a location. About midway down the page, where I've got a blue box outlined in really teeny tiny font, that's where you'll find all the values that you're going to need for K. So now you know the secret because you attended this conference. You know the secret to finding K. I did transcribe some of the values here on my slides so we could take a bit of a closer look at them. First, I'm going to introduce a unit, the endotoxin unit. That is just a measure of the biological reactivity of the endotoxin, and you'll see K expressed in endotoxin units per kilogram. But the other thing that we notice about K is that we've got a lot of different values for it, right? So there's a K for intrathecal injections. There's a K for injections by all other routes of administration. There is a K for products dosed based on body surface area, and we see a lot of our oncology products dosed in that way. There are also a couple of other Ks for radiopharmaceutical products that I didn't list here, but again, I've highlighted something on the slide, and that is the 5 EU per kilogram K. We're going to do some math here in a minute. Don't act too excited. We're going to do a couple of calculations here in a minute. So you're going to see that 5 EU per kilogram again. So I want you to remember that. Now M is much more intuitive, right? It's just the maximum dose of your product. That's pretty straightforward. But what I want to caution you when we're talking about M is that you need to take the value of M from your product label. Now, John mentioned this earlier in his talk as well. You cannot guess M. You cannot take M from a monograph. You need to take M from the product label. One other thing that we don't see as much, but I wanted to add it on the slide, that is if you've got a kit, so a product that's co-packaged, maybe product and diluent packaged together, the endotoxin contributions of both of those should be considered as one, but we don't see that too terribly often. All right, so you see that both K and M have aspects of body weight. So what weight are we talking about here? For endotoxin calculations, OPMA considers the standard adult body weight to be 70 kilograms. But we know that there are products that have indications for a pediatric population as well. And those need to be considered when you're setting that acceptance criteria. So to find the appropriate pediatric body weight for your patient population, we recommend referring to the Center for Disease Control's weight for age charts. And I'm going to show you how we approach that in one of my case studies here. Now, if you're setting a specification and you feel like you're doing a lot of legwork, a lot of calculations, had to figure out, my M for this population, all this stuff, there is a place in the submission where you can explain your thinking, you can explain your rationale. And that's in section 32P56. It's a great place to discuss the rationale for your K and for your M. Okay, case study time. Time for me to do math on stage. Everybody ready for that? Yeah, I had somebody check my math beforehand, so I had a reviewer actually check my math beforehand. All right, we're going to do one simple and one complex case study. So we'll start out with the simple. The product for our simple case study is called leuprolide acetate. It is a product for the treatment of prostate cancer. It's got a very simple dosage information on its label. And this is pretty much all it says. The recommended dose is 1 milligram administered as a single daily subcutaneous injection. There's not much there, but we've already got enough information to figure out how we start to set up the problem for our acceptance criteria. So this is a subcutaneous injection, not intrathecal, so we know what our K is going to be. There are no pediatric indications listed on this label. And finally, this is just a vial of drug product, no kit to have to worry about. So we go back and stare down our acceptance criteria calculation that we have to do. But hey, we have all the information that we need. We take our 5 EU per kilogram K. We know our dose and our adult body weight. And so after that, it's just a matter of doing a little bit of arithmetic and canceling out the units to get an endotoxins acceptance criteria of 350 EU per milligram. Easy peasy. Okay, now let's turn it up a little bit, make it a little bit harder. The product for our complex case study is a product called levetiracetam. And this is an anti-epileptic, present in a lot of different formats, tablets, but there is also an intravenous injectable format. Okay, that already tells us something. We already know that our K here, because it's an intravenous injection, is going to be 5 EU per kilogram. Ooh, but look at that dosing information. That is a mouthful. We've got pediatric dosing, we've got lots of different ages, lots of different doses. Well, okay, so we're gonna have to look more closely at this. Now, obviously, I've worked this example for us, and I'm not gonna go through this line by line, but hey, I think we should take a look at this one as maybe being our maximum dose for the drug product. Got patients 12 years or older with a dose of up to 1,500 milligrams. All right, so we're going to turn to our CDC weight for age charts, 1,500 milligrams in a 12-year-old patient. So these charts separate male and female, and they also do the age by month. So what we're looking for here is we're gonna look for a 144-month-old female. Why female? The weight is lower, so it gives us a worst case for the dose. We also recommend using the 50th percentile weight for this calculation. So what we end up with here is a 41.8 kilogram patient. Do our math, 36 milligram per kilogram. We're thinking that that is our worst case dose. So I go back to the label, just double check, and yes, 36 milligrams per kilogram is our worst case dose, and now we have our M. We had to work for that M, but now we have it. Plug in our values, do our arithmetic, cancel our units, and we end up with an endotoxins acceptance criteria of 0.14 EU per milligram. All right, couple of resources for your perusal. USP 85, I talked about that the entire time really, but one thing I want you to know is that there is one type of injectable product that doesn't have its K in 85, and that is ophthalmic injections. You're gonna find that information in USP 771. Got our CDC weight for age charts. But honestly, you could just Google CDC weight for age charts and that information will come right up. There's the URL if you need it. A great guidance on endotoxins testing and a guidance on our controlled correspondence program, where again, we do get a lot of questions about that endotoxin specification. Okay, recap. Exposure to excessive endotoxins is a patient risk. Process design and in-product testing are both important things that work together to ensure that endotoxins are controlled in your drug product. USP Chapter 85 contains information regarding both methods and acceptance criteria for endotoxins testing. And when you are setting that acceptance criteria, you should be taking your information from the product label. Okay, time for our quiz, and then we'll wrap up for the day, and I'll turn it over to our next speakers. Okay. Now, you're all very clever, I know, so I know you're going to get this. The acceptance criterion for the endotoxin specification for a subcutaneous injection should be calculated from the formula provided in USP 85. I don't hear anybody. That's true. As I said, you guys are all very clever. Excellent job. Next one is much less wordy. Okay. K is the same for all injectable drug products. True or false? False. Oh, you guys are great. Excellent. All right. Well, that is the end of my time and my material today. So thank you for your kind attention, and I'd like to turn it over to our next presenters. We've got Bailey and Sunny. Thank you all very much.
Thanks, Erica. Good afternoon, everyone. We're going to switch gears and do a little policy and labeling talk. So my name's Bailey Larson. I'm in the Office of Generic Drug Policy on the Patent and Exclusivity team. I'm here today presenting with my OGD colleague, Sunny Pian from the Division of Labeling Review. We're going to talk about ANDA's labeling requirements for RX to OTC switched products. Before we begin, just want to go over the learning objectives. We'll explain how a prescription or RX drug product may initiate the change in marketing status to over-the-counter or OTC through the new drug application or NDA process. We'll discuss the process for labeling and policy updates when a reference listed drug or RLD undergoes an RX to OTC switch, and identify the roles of the agency and the roles of you all, the applicant. So just want to read the room and online a little bit. There'll be the poll online and we'll collect the results. I won't be able to see them live, but how many in the room have you sought approval for an RX to OTC product? Anyone, yes? Okay, and I'm assuming the rest are no or with the FDA. So thank you. We've got a couple, and hopefully you will learn something, and if your RLD ever makes the switch in the future, you can remember some of these tips. So as an overview, there's two types of RX to OTC switches. There's a full switch and a partial switch. The full switch is when an NDA holder switches the drug product covered under the NDA to OTC manufacturing status in its entirety. So in other words, all the conditions of use. When a full switch occurs, there is no longer the prescription product. It's only marketed as over-the-counter. We'll be focusing on full switches for this presentation. A partial switch is when a NDA holder switches some of the conditions of use while retaining others with a prescription status. This usually requires a full new NDA, and the product will have different conditions of use. So examples of this are levocetirizine solution or proton pump inhibitors. I'm sure we've all seen that PPIs can be over the counter for short-term GERD or prescription status for things like peptic ulcer disease. So we'll focus on full switch tips today. So how does a full switch come about? The NDA holder will switch the drug product to non-prescription marketing status in its entirety by submitting an efficacy supplement. We've talked a lot about PAS today with David and John, so the similar things. There was a change in recent years in the agency that the RX and OTC product are now considered the same drug product, so they'll even have the same product code. NDA holders may submit patents within 30 days from the date of approval of the efficacy supplement. And holders or applicants of approved and pending ANDAs referencing that NDA must submit a supplement or amendment to their ANDA relying on the RLD for OTC conditions of use and associated labeling. So that's really the key bullet here for this audience. It does not require a completely new ANDA, which as we know is beneficial. There'll be no unnecessary data collection, quicker review times, and no user fee agreements. So here's some high-level regulations. After a supplement is approved, the FDA will continue to list the patent information that was listed for the original NDA for 30 days or until Form 3542 is submitted. If the patent was already listed in the Orange Book, the patent submission date will stay the original pre-switch date. If patent information is submitted, the date for the patent information will reflect the original date of submission. Some examples of this are a relisting of method of use codes, adding a new patent to cover the new conditions of use, or a revised MOU. If the NDA holder does not submit Form 3542 within the 30-day period, the carried over patent information will be removed from the Orange Book at the end of the 30-day period. Because the OTC and RX product are now considered to be the same listed drug, that also applies to 180-day exclusivity. So why do we care about this, and what does it matter, and kind of what's the history behind all this? So it all goes back to the patient, right? We need to protect the public. In the 1940s and 1960s, there was abuse of amphetamines and barbiturates, which was one key factor that led to the Durham-Humphrey Amendment of 1951. So this act was what distinguished the OTC and RX drugs. Prior to this, the determination as to whether a drug was safe and effective for self-medication was left to the manufacturer. So as you can imagine, this was really confusing for patients and pharmacists and other healthcare providers, and it limited the agency in deciding what was safe for over-the-counter use and without medical supervision. So this amendment in 1951 gave the agency regulatory power. This is also where the labeling of RX only came from, so we've all seen the caution federal law prohibits dispensing without a prescription. And when things like this break down, we get misbranding. So RX and OTC versions of the same drug product are not permitted to be marketed at the same time. We must have a reasonable difference, so either dosage or indication or route of administration. Failure to maintain labeling that is consistent with that of the RLD is basis to propose withdrawing approval of an ANDA. So I'll now bring up Sunny to tell you a little bit more about the labeling requirements.
So thank you, Bailey. So again, my name is Sunny Pian, and I'm from the Division of Labeling Review. And I'm going to kind of go over what this means actually for your ANDA application. So first, we're going to discuss pending original applications. Pending ANDA, if there is a RX to OTC switch, you must submit an amendment to conform to the labeling requirements. So if new patent information for the OTC entry compared to that of the prescription entry is listed, that is, newly listed patent or revised method of use, and the applicant must provide an appropriate certification, recertification, or statement. So I'm going to provide you with the hypothetical, just kind of example. So let's say that there is a drug, and we're going to call this drug Dendrocalm. This is completely made up, so you don't need to look it up. So let's say for this drug Dendrocalm, it has the the prescription or the RX indication, and the indication is for the treatment of the fear of forest. So you're definitely afraid of the forest, and has 3 patents associated with it. Patent number 1 is for the treatment of the fear of trees. Okay, patent number 2 is a treatment of the fear of pine trees. Okay, and then patent number 3 would be for the fear of bugs, because we find bugs in trees. So now the NDA submits an OTC switchover, and let's say for the OTC indication, it's approved or they're proposing to treat short-term fear of trees. So as you can see, with this indication, it's a more limited indication, but it's still overlapping. So the first indication, so the treatment for the fear of the forest, the first RX indication, no longer exists when it switches over to the OTC product. So the NDA applicant or the NDA sponsor, let's say, submits a new efficacy supplement for a full switch to the OTC. So patents 1 and 2, so again, patent 1 was the fear of for the treatment for the fear of trees, and number 2 was for the treatment of fear of pine trees. Okay, so patent number 1 and 2, they get carried over, and the use code stays the same. At this point, the ANDA applicant must recertify. Now, for patent number 3, which is fear of bugs, it is not carried over as it is not related to the OTC indication. So the NDA, with the OTC supplement being approved, the patent and the use codes are it must be addressed by the ANDA. So again, patent numbers 1 and 2, which has the overlapping indication or protection for treatment of fear of trees and pine trees, they must be certified or recertified. Okay, so now that we discussed the pending originals, we will discuss approved applications. So ANDAs are responsible for monitoring the Orange Book for updates to the RLD. So approved ANDAs is required to submit a prior approval supplement, a PAS, to comply with the updated labeling requirements. So generally, an ANDA labeling must be same as your RLD, with the exception of things that may be due to manufacturing or distributor differences. And again, along with monitoring for the Orange Book, it is also the ANDA applicant's responsibility to monitor drugs at FDA to make sure that you submit supplements or amendments accordingly when your RLD updates. And again, specifically for the RX to OTC switches, the ANDA applicant must submit a PAS as soon as possible. Otherwise, your ANDA is considered to be misbranded. So when you do submit your PAS or your amendment, but in particularly for the PAS, a filing review and subsequent acknowledgement supplemental ANDA receipt letter will be issued with the appropriate goal date attached to it. And this PAS will undergo standard review practices. OGD will then issue any formal communication through our standard practice through the IR and the DRL, so information request or discipline review letter process. So now I have challenge questions. So for challenge question number 1, which of the following statement is not true? Is it A, RX and OTC versions of the same drug products are permitted to be marketed at the same time? B, pending ANDAs should submit an amendment to switch their ANDA drug products from RX to OTC. C, ANDA applicant is responsible to update their labeling. And D, FDA does not consider post-full switch NDA product to be a different listed drug. Any ideas? Yes, thank you. So the correct answer is A. RX and OTC versions of the same drug products, it says are permitted, but they are not permitted to be marketed at the same time, as Bailey has discussed in her presentation. Okay, so challenge question number 2. What type of submission should approved ANDA applicants submit for a full RX to OTC switch? A, a CBE 0, B, CBE 30, C, a PAS, or D, submit a new ANDA? Correct, thank you for paying attention. Yes, a PAS. You will be submitting a prior approval supplement. So in summary, pre and post full switch drug products are considered to be the same drug product under Section 505J 5B4 of the FD&C Act. ANDA applicants are required to update their labeling to be consistent with the most recent approved RLD labeling. Pending ANDAs must recertify to any new or updated patents from the RLD. And lastly, monitor for RLD updates. When an RX to OTC switch is identified, submit a PAS or an amendment as soon as possible. Thank you, and then now I will pass it over to Captain Vincent Sansoni and Commander Rinku Patel for their next presentation.
Good afternoon, everybody. I hear this is the last presentation, so I'd like to say they saved the best for last, but given everybody that went before us, I don't think that's appropriate. But anyway, hope this helps. And I think this presentation is apropos because this is actually regulatory reminders to the finish line. So this is actually kind of wrapping up the day, which makes sense because this is those later stages where everything's going to be kind of wrapped up and moving towards approval. So we're hoping to get that action. So I think it's a good way to cap the conference and close it out. So myself is Captain Sansoni. I represent the Division of Project Management. With me today is going to be Rinku Patel. She represents the Patent and Exclusivity team. And we thought it would be befitting to do this presentation together because the Division of Project Management and the Patent and Exclusivity team actually work hand in hand when it comes down to these final stages to approve an application. So we thought both groups together would be adequate to present today. So when we talk about regulatory reminders to the finish line, what we're actually talking about is finishing up those last stages to approval. So your application has made it through those rigorous scientific reviews. You know, you're moving into the final stages. Your deficiencies have been deemed adequate, and you're looking at getting that application right next to the finish line, getting that approval letter that you want. So that's kind of where we're kind of at right now and what we want to monitor in between that. And you'll see us kind of reiterate the word vigilance and monitoring throughout this presentation, because this is really where you want to, you know, don't lose gas, right? You want to step on the pedal here and make sure you're watching things that will come over today to make sure we get that application to approval and it gets to you as soon as we can. And I guess it's important to also note that, you know, we have a shared interest, right? The FDA and industry to get that application to approval. We want to make sure that we facilitate, you know, generic entry just as much as you want to get that application approved for the same reason. So we kind of have a shared interest in this category. So we definitely need to work together. It takes the village, and that's what we need to do. So what are some of our learning objectives for today? We're going to discuss how and when to respond to changes to patent information. And we need to be very cognizant of the fact that these changes to patent information occur throughout the life cycle of an application, right? It's not just when you submit the application that these changes stop. Changes occur throughout the life cycle application because you have a lot of confounding variables that are happening outside of your control as well. And again, you need to monitor those changes. We're also going to review the best practices for ANDA applicants. So what to do when these changes arise, what to do when these changes occur, how to address them, what's the best way to get it done, things like that Commander Rinku will go into and give you a little bit more detail, and that'll get you through the next phases of getting this application to approval. And then lastly, we're going to bring it all together. We'll go over some other things that you need to be vigilant and cognizant of to monitor for your application, and we'll wrap it up all together, hopefully so that you can see the bigger picture and how this application is going to get to its final stage. So we didn't want to step off the gas here a little bit, and we wanted to also talk about what is industry doing well. Well, industry is doing well at the beginning of the submission, and the pretext of this slide is all at the submission of your application. We find that these 4 bullets kind of being addressed during the initial stages of your application when you're submitted at filing. You guys are the industry certifying to all patent listed, doing a great job there. Please keep that, keep that going. You're providing timely notice. That's good. You're notifying the NDAs, et cetera, of your patents, patent certifications. You're submitting the documentation of notice for original P4 certifications that we have to have as a regulatory requirement. So you're submitting those documentations of notice on time. And you're also notifying the FDA of the first civil action complaint, meaning, hey, if you guys got sued or industry was sued when you submitted these notices, you're telling us of that fact, which is good. But this is all on the onset of the application. This is at the time of filing, which you're doing a real good job of. But we want to make sure you guys understand that this not only stops here, this also matriculates through the application process, meaning you're going to have to, monitor for the patent changes to the Orange Book, et cetera, and things like that, and make sure you're meeting all the necessary regulatory requirements that ensue. So it doesn't just stop when you submit the application, it goes all the way up to the final approval. And again, we're at the final approval stages. We want to help you break through that barrier, make sure you're not missing anything, not have us kind of try to chase you down to make sure you submit necessary regulatory documents that would hinder approval. That's where you're going to get stymied. So I think Commander Rancu is going to go over some of these specific details of where we've seen applicants might need a little help and a little bit more information.
Thanks, Vince. As he said, in the next 3 slides, I'll be sharing common deficiencies and sort of observations that we have, as well as identifying areas of improvement. So going to the first bullet here, addressing timely submitted later listed use codes. So you might be asking yourself, when can that actually happen, right? When can an NDA holder submit a new use code, or what could trigger an NDA holder to submit a new use code that for a patent that's already been listed in the Orange Book? There are a handful of instances where a newly listed use code can be submitted and to be considered timely listed. A common situation is when an NDA holder has obtained supplemental approval seeking a new indication, and they submit a updated patent information within 30 days of approval of the corresponding labeling change. So essentially what you would have to do if the patent is submitted or the use code submitted to and is considered to be timely listed, you've got 3 options. You're either going to submit a Section 8 statement indicating that you're planning to carve out that indication and sort of initiate the carve out process, or you're going to be retaining the protected information, and in which case you've got 2 options. You're either going to state that you're going to wait until the patent expires and submit a paragraph 3 certification, or you're going to challenge the patent and submit a paragraph 4 certification. Either way, you must be consistent across all patents that are associated with that use code or similarly worded use codes. What we often see is applicants trying to take the original paragraph 4 certification and state that they have already addressed the patent and therefore the use code with their previous P4 and notice. However, we require you to submit new P4 certification. Simply put, a previous P4 certification and notice associated with that previous certification cannot cover a new use code that was submitted after the original notice was sent. Therefore, it's not acceptable to address the use code without submitting a new certification. And essentially, by the virtue of a P4 certification, you have to send a new round of notice. Some of the common challenges that we run into is sort of showing adequate documentation of notice. And there are times where you guys do really well and you guys submit, you've addressed the use code with a P4 certification and you've noted and you've sent the documentation, but you just haven't told us about it. So when you've done the first half of it, be sure to submit the other half of it so we know that you've done that. And the other sort of instances where we often see is that notice itself is not aligned or incomplete with the requirements under 31495, the notice requirements. So be sure to ask yourself, have I checked to see if the right applicants got the notice? Was the notice sent to the right address? And more importantly, was the notice actually delivered? So transitioning on to exclusivities, when exclusivity is recognized for the relied upon listed drugs such as 3-year exclusivity or orphan drug exclusivity, there are 2 potential pathways. You either are going to retain the protected information in your label, or you're going to omit the protected information and seek to carve out. And you're essentially saying if you're going to be carving out that information, you're seeking approval of the application prior to the expiration of that exclusivity. Either way, just make sure you're clearly stating the intent of how you're going to be addressing the exclusivity. And to the extent that you have a licensing agreement, you've procured something to say that you are able to go to full approval with retaining the protected information prior to the expiration of that exclusivity, you must obtain a selective waiver. And in that waiver, what we're looking for is it on NDA letterhead? Does it indicate the effective date? And to the extent you can, ideally, we would want to know what exclusivities has the NDA holder, or what's the agreement for? What exclusivities has the NDA holder essentially wait for you? So we all know folks are eager to get their applications approved, and so are we. We want to help you guys get to the finish line and get the application out the door. However, it's imperative for you to submit litigation status on patents that give rise to a 30-month stay. Otherwise, we'll assume that the earliest lawful approval date is the expiration of the 30-month stay. And the regulations do require that an applicant under 314-107 does require the applicants to submit within 14 days of the decision that's been entered by the court, submit that information to the agency. And so for all the information, for all the hard work that you've done and that you made sure that you've got the litigation terminated, just tell us about that, and tell us about that so we can help you navigate what the earliest lawful approval date is. One area that we sort of run into as well is whether or not the order itself was signed by the judge. What we can't do is assume that the proposed stipulation in order is what you guys have agreed to, because it's possible that there's another agreement that happened after you have submitted the one you've sent to the FDA. So what we want to know is it signed by the judge? Is this the order that terminates the 30-month stay? And additionally, the regs do require that the order must be signed. So again, providing us with the litigation status helps us determine what the earliest lawfully approvable date is for your application. Now shifting gears to sort of post-approval requirements. Commercial marketing requirements are required for both sort of the traditional patent challenge 180 exclusivity for those who were considered to be first applicants who have challenged a patent, or for first approved applicants who satisfy the definition of competitive generic therapy in the exclusivity sort of provisions. So to highlight some key points in relation to the traditional 180 exclusivity, commercial marketing notification must be submitted in order for us to know that you've triggered your 180-day exclusivity. Not only does it benefit you, it will also make an impact on any other approved applicants to indicate whether or not they have to sort of initiate, or they essentially know that they've triggered, that 180-day has been triggered. 180 triggered, 180-day exclusivity, any triggering of that also impacts any unapproved products as well. By the definition and sort of what the statute provides is that commercial marketing, not only of your drug, but of a listed drug, so that means authorized generic, you must send that information to us as well. And then to the extent that you are a first applicant that was approved, but you did not secure final approval or tentative approval within the 30-month time, and what we did is we punted on your 180-day exclusivity, it's possible that you're still eligible for that 180-day exclusivity, but you have to, looking at the approval letter, you have to indicate that you've actually commenced marketing. So to avoid losing the benefit of part of the 180-day exclusivity, you must submit this information within 30 days of when you've commenced marketing. If you do not submit this information within 30 days, the date of approval of your application will then govern the date that the 180-day has been triggered. I'm going to say that again. To avoid losing the benefit of the 180-day exclusivity, you must submit the required documentation within 30 days of when you've commenced marketing of your application. If you do not submit this, the date of approval will then govern the date the 180-day has been triggered. And sort of shifting gears to CGT exclusivity, the blocking effect for folks who are sort of in this arena and have had an approval letter, you will indicate, which indicates sort of in length what the requirements are. We will not be blocking any other approvals from happening until you've triggered your 180 exclusivity. So it's very imperative for you guys to submit that information to us. The letters say that they have to be submitted to your ANDA file, as well as indicating and letting the patent and exclusivity team be aware of that in order for us to prevent or halt any other approvals, any other subsequent approvals. And the forfeiture space for CGT exclusivity is very simple. If you don't commence marketing in 75 days, you've forfeited your exclusivity. We try to provide as much transparency as possible. The Paragraph P4 website and the CGT websites both indicate the eligibility of forfeitures or the eligibility of 180-day exclusivity and whether or not an applicant has forfeited, as well as indicating when they've commenced marketing. So, Vince, I'll turn it back to you.
Okay, so why am I back up here? Well, we've walked through all the patents, you've gotten everything straightened out, you know, you're now in those final stages. Well, you want that letter from the Division of Project Management saying, hey, my application is approved, right? That's the end goal. That's the goal of all of us to get this application. Well, we still want to reiterate, just because you've, listened to Renku, you got our presentation down, that's not where it stops. Monitoring is paramount, and it's paramount throughout the life cycle of the application. There's other things you want to make sure you've been vigilant of. Some of those things, just to make reference here, USP updates. Are you conforming? Make sure your facilities are in line. Did any of those facilities become withhold, or there's a problem with the facilities in the late cycle of an ANDA that could derail it? You want to make sure that you monitor the Orange Book, and you want to make sure you monitor drugs at FDA for RLD updates. You need to monitor these things. If you monitor these things and act accordingly and in an expeditious manner, you will get to that final finish line for approval. So again, monitor. If we ask, anything of a Reg Affairs department externally, it's be vigilant, make sure you stay on top of things, stuff that are outside of our control. NDAs are submitting supplements to their respective applications all the time. Are you modeling the labeling accordingly, et cetera? All those things need to be monitored by the external stakeholders, industry. So we want to make sure your Reg Affairs department are doing their due diligence. That way you can help us, and we get to that same overarching goal to get that application out the door. So that's kind of where we're wrapping up. We'll go over some challenge questions now. So the first challenge question is, within how many days are you required to submit notification of court actions or consent to approval? 60 days, and we'll go through the end. 30 days, 14 days, or 10 days. This is where I would usually go around the room using the Socratic method, but I'll spare you guys since the room is so empty. Answers 14 days. So that's the time you need to make sure all your court actions and consent to approval are sent. Challenge question 2. Which of the following statements is not true? I can be eligible for final approval before the 30-month stay expires without having to submit a court action. A court order does not need to be signed by the judge. There is no set requirement to submit a documentation of notice. Lastly, my original P4 certification will cover a newly listed use code. I hope nobody guessed B, because I think that was our easy giveaway. But the correct answer is all of the above, right? These are not true. So we want to make sure that you guys do all these things in a timely fashion. Rinku went over a little bit in this presentation. Submit those notices, submit the documentation, submit the necessary regulatory requirements, and you'll get your application to approval. So that's it for our presentation. Thank you guys, really appreciate it. Right now, I think we're going to turn it over to Brenda for the Q&A panel.
Can we have 6 speakers come back up to the podium, please? As we are getting settled in, I do have a couple announcements, and it really pays to stay to the last. Okay, let's start with the online questions, please.
Okay, first question is for John. Can a bioburden sample between the pre-sterilizing filter and sterilizing filter be justified and accepted by FDA if the filtration process is lineal without holding periods and the substance has antimicrobial properties like antibiotics?
Oh, there we go. So you're asking if you can still sample in between filters. I just want to make sure, I'm having trouble hearing you. To make sure you can still sample in between filters if there's other situations going on with the drug. I think the answer is no.
We want sampling. If that
we want
helps.
sampling prior to filtration.
Between pre-sterilizing filter and sterilizing filter.
Prior to filtration is what we're looking for.
Antimicrobial properties of drug products shouldn't be relied upon to improve your CGMPs, right? Like you need to treat drugs kind of the same in that way. John and I work together, so
It's a package deal.
this is how we spend our days. So
Okay, thank you. Next question, please.
Okay, next question will go for Erica. Please elaborate on the CDC weight table in a little more details. For example, what is a 50th or 75th percentile weight and why 50th percentile is to be chosen?
oh, that's a good question. So I don't know if you've had like little children that you have to take to the doctor and the doctor's like, oh, they're the 99th percentile of the weight. That's what those charts are essentially the same chart. That's probably what your doctor is using. It just means that I think the actual thing that it means is it encompasses 50% or 70% or whatever the number is of the population. We choose 50 just to give sort of a safety margin for individuals who may be smaller than the highest weight, but also to still be reasonable with endotoxin specification setting activities for pediatrics. Now, I would say there could be a scenario where it would be wise to choose a lower weight, to choose a lower percentile, choose something like the 25th percentile, depending on the patient population that you're talking about. So if you've got a patient population that's very dramatically underweight, you may want to go lower with the weight that you choose for those calculations. So, John, you want to jump in on mine like I jumped in on yours?
You know what? I would just say also justify it. If you make a choice, explain it. If you explain it with some reasonable thought behind it, we're probably going to agree with you.
Okay, thank you. Let's go to in-house question, please.
Hi, my question is to John. So sometimes when we're using ready-to-use container closures like stoppers and vials, do you think it's sufficient to just reference the LOA, or do you also need like a statement from the company?
So I assume you're talking about referencing.
The sterility assurance package.
Yeah.
Yep.
So you would need an LOA.
Okay.
Are you talking about a DMF, I assume? Or
Yeah,
are you talking about
DMF.
sterilization or deprogenation of these components?
No, if you're not deparenate it and we're just using a ready to sterilize or ready to use thing, do you think the LOA would suffice?
Well, the LOA will give us access to the DMF, which has all of the information in it.
Okay.
So I think just have that LOA as clear as possible. Have the company tell, because we understand you don't know those details, right?
Right.
The company should be able to tell you, here's what you purchased. That information is in volume 14.
Okay. And I also have a second question to that. So sometimes we do have like container closure DMF owners who make changes to their sterility assurance package, and they also notify us of the changes, but we don't have like a lot of details on it. So do you think that we need to take the more conservative approach of like having a supplement change, like a CPE 0, or should we just put in a control correspondence in situations like that?
That's a good question. So you're talking about the DMF holder is making a change
Yep.
to their sterility information. I believe what happens is they tell you, right?
They
And then you
did.
file if it depends where you are, if you file an amendment or a supplement for that change, it would, right? And then it would come to us that way. So yeah, we would still need the information. I mean, enhance the review, right? If you don't tell us where it is, we'll probably find it. It just won't be as efficient.
Okay, but in this situation, you think that we should take more of a conservative approach by putting like an annual report or like a CVE 0?
No, I would wait. I mean, again, it depends on the change, right? Because some
Yeah.
are annual reportable, things like, there's a lot of complicated situations
Yeah.
going on. But I think I would just listen to them and I wouldn't just start bombarding us with potentials. You know, like it might have changed or there's an annual report, then it might not even get assessed if it's an annual report. It's convoluted.
Okay.
Okay, thank you. Let's go to online question, please.
Question for Bailey. What happens to the RX ANDA once the associated RLD undergoes a complete switch to OTC? And how long can the generic manufacturer continue to market to the RX ANDA?
Can you repeat it?
Sure. First part, what happens to the RX ANDA once the associated RLD undergoes a complete switch to OTC? And the second part is, how long can the generic manufacturer continue to market to the RX ANDA?
I think the clearest answer is, in general, the ANDA needs to submit the supplement as soon as possible. As soon as they get notification of the switch, they should be submitting the prior approval supplement to match the labeling exactly of the RLD.
Okay, thank you. Next question, please, online.
Okay. Next question is for Rinku. How and what should an ANDA holder communicate to the agency when they believe they are still eligible for the 180-day exclusivity despite not obtaining a TA in 30 months from the time of submission?
Okay, so I'm just going to paraphrase the question. So the question is, what should I submit to the agency if I did not secure tentative approval within the 30-month timeframe, but I believe that I still hold exclusivity? So I think the simplest format would be to submit some sort of correspondence indicating that you have, for your reasons why, and the basis as to why you think that the 180-day has not been forfeited.
Thank you. Do we have more online questions?
Yeah, next question will go to Sunny. What is the best way to know if my RLD has updated?
Hi, thank you for that question. The best way is just to be vigilant. You can check Drugs at FDA, or also there is an email listserv that you can go on the FDA website and sign up for to receive updates.
Thank you. We do have a question in-house, please.
Hi, this is kind of again, thank you all for your presentations. It's kind of a follow-up to that online question. So does an ANDA have a time that they'll be able to leave their product on the market as an RX when the RLD is switched to OTC unexpectedly? Or do they have to somehow withdraw product because its labeling has significantly changed from an RX to a OTC?
So I can take that. So when it does switch over, you do have to submit, and you do have you do have to be same as your RLD. In terms of what you do with what's already out there, that is a business discretion that you would have to do. You would have to make, basically.
Okay. Thank you. Online question, please.
Question for Erica. If we are doing container closure integrity testing in lieu of sterility for stability testing, can we omit endotoxin testing from stability and just do at the release?
So John, you can jump in on this one too. I'll take the first whack at it. All right. So what the questioner is asking about is there is the ability to, as part of the stability program, instead of doing sterility testing, to instead test the integrity of the container closure system, which kind of makes sense, right? Like over time, where's the sterility failure going to occur on the shelf? It's going to occur because the container closure system degrades. So that is allowable. So it is also allowable under some circumstances to omit endotoxin testing at particular time points, but we recommend certainly thinking about that, making sure that it's the correct decision for your product, and keeping endotoxin testing at the final time point for the stability program.
Thank you. Next online question, please.
Question for Vince. I'm a U.S. agent that recently started working on ANDAs. Could you share three key recommendations that would assist with my application moving forward to approval?
So yes, I can. I don't know specifics because that's a broad question. There's many things, but if I was a regulatory agent that was just starting, and I would say again, you want to make sure you have all the necessary documentation in there, meaning you've been keeping track of your ANDA for one, meaning you've made sure all the responses were submitted to any kind of IRs, DRLs, and CRs. That's first things first. Make sure that you're keeping up with your communications. Second thing, like I mentioned earlier in the presentation, was monitoring. You want to make sure you're monitoring the necessary external changes that can affect DMF updates. You're staying in contact with your DMFs, making sure that they're not submitting any amendments that you don't know about that could potentially derail. That's another kind of regulatory thing that I think is valuable that's overlooked. And the last one is definitely probably making sure that you're staying in touch with your facilities, making sure they're up to date, making sure they're all adequate and nothing has transpired on that front that would derail the application.
Thank you. Online question, please.
Question for Rinku. What happens when there are multiple first filers eligible for 180-day exclusivity and one of the first filers does not provide notification of commercial marketing within 30 days? Will this impact the start of the exclusivity for all the first filers?
So I'm just going to paraphrase the question here. So the question is, what would happen to that applicant's 180-day if they do not submit notification, even though there's other approved applicants in that space? So assuming that you've actually commenced marketing, because the question is a little bit more vague than originally indicated, you would be essentially triggering 180-day exclusivity for the entire cohort, and maybe the other applicants will sort of note that too and want to, there's an incentive for you to submit it. There's an incentive for the other applicants to know about it. Therefore, they can start triggering their marketing as well.
Thank you. Next online question, please.
Right, question just for the panel. When an RLD undergoes RX to OTC switch, but the ANDA holder does not switch and continues to market the drug as RX and appears on an orange book as RX, does the agency monitor this, and what action will the agency take considering the product is deemed misbranded?
So just to, I guess, rephrase the question, basically what happens when an ANDA applicant does not convert over, correct? So basically, it would really just depend, our favorite answer, but initially, we would have to kind of see when the switch happened for the NDA, because if we're talking just maybe a week or two, then, we got to give people time to also submit, right? But we do monitor, but if we are aware of it, we do monitor, and if compliance needs to take action, we will go that route.
Thank you much. That's it for online questions. Anyone else in the house with a question, a very burning question? Doesn't have to be very. Any questions? Okay, well, that brings us to the end of the session, and we thank our panelists, but let's hold on. We do want to invite Tony Schumer, who is the Senior Advisor from the Office of Generic Drugs, to give us her closing remarks and wrap up. Would you like to stay seated? Okay.
Thank you, Brenda, and thanks everyone for being here. Just want to wrap up today with a summary of the forum, and then share summary of the forum. fun conversation with someone who was talking to me about our usage of acronyms. So to be honest, we do our best. We definitely want to be mindful of using acronyms. It is like a second language to us, but even in-house, we get a little lost and confused in our own acronyms. So we do our best to explain what those acronyms are in the beginning, but in our daily language, those acronyms definitely sneak out on us. So in closing thoughts, what I want to leave you with today is a few tips. Every product that you're developing, every submission that you're preparing, and every interaction that you have with FDA has its own unique elements. So to make sure, to help you out with that, make sure that you know what the resources are available to you. My call to action to each and every one of you is there's so many resources out there, and because of the unique elements of every situation, create a customized toolbox to help you. When building a customized toolbox, it's really important to have a strong regulatory affairs department to help you with that. Don't forget, when creating your customized toolbox, the GDUFA 3 commitment letter is also a valuable resource. The GDUFA 3 commitment letter provides new enhancements to the program, which are designed to maximize the efficiency and utility of each assessment cycle. It's intended to reduce the number of assessment cycles. So these new GDUFA 3 enhancements offer a lot of opportunities for prospective applicants, and application holders to interact with the agency more than any of the 2 previous iterations of GDUFA. While our presenters may not have specifically referred to the GDUFA 3 commitments with every presentation and every tip that they provided to you today and yesterday, be sure that they did and refer to these opportunities often. We understand that there's a lot of information, and to ensure that the pathway to generic drug access continues, we want to be sure that you have the resources you need and you know how to use them. This is where that customized toolbox comes into play. We also understand that all of this information is hard to process. This is why we participate in events like this one. As you heard from Brenda and others throughout the forum, FDA participates in several forums, conferences, and we also prepare a lot of webinars on current issues to help provide you with these resources. We also like to share best practices and current tips for you. I think I even heard about secrets today, so this is the advantage of attending these forums. Please keep an eye out for all of our events, as they might help you with developing this customized toolbox that I'm speaking about. We look forward to seeing you all at our future events. If you were here yesterday, I believe Brenda started the day off with wanting to know how many SBIA events that you have attended. I think she was a little disappointed when she didn't see more hands of folks that had attended to 10 or more. So this year alone, I think that they have probably at least 10 events. So hopefully you guys can keep attending those, and next year you can help Brenda out by making her feel better that you have participated in many more. We do a lot more outside of SBIA. We host our own events. We also work with the CRCG. See, here I'm using an acronym. The Center for, does anybody remember what that is? Center for Research of Complex Generics. So keep an eye out for those. In closing, I want to thank each of you for participating in our first hybrid GDF since the pandemic. Whether you were here or in person, your participation demonstrates your quest for knowledge and desire to stay up to date on current topics. We hope the event was useful and informative. Before I walk away today and before we all leave with our free parking tickets, I want to give a big thanks to SBIA for hosting this event. Brenda, Will, Jeff, Nora, Matt, they're all in the back of the room. So if you could please turn around and give them a wave and of appreciation, we would really appreciate that. Thank you so much, everyone. Drive home safe.